Okay, hi everybody. I am Diana Milowitz. Just a disclaimer, there'll be no Superman or even Wonder Woman in my presentation. So I am in the division, in the Department of Medicine. I direct the Division of Medical Genetics. My offices and labs sit right in the middle of biochemistry, so it's sort of natural that I have a cross appointment into have an appointment in the biochemistry and cell biology graduate program. I got my MD PhD from UT Southwestern and did my PhD with Brown and Goldstein, who were awarded the Nobel Prize in Medicine for discovering the LDL receptor. And that really, their work really illustrated for me, working with them really illustrated to me how powerful human genetics can be in terms of identifying genes that predispose people to human diseases. So I trained in internal medicine and cardiology and genetics. And when I was a fellow, I saw a patient with Marfan syndrome. This was a condition. It is a condition that causes people to grow very tall. They have long arms and legs. And in addition to that, they have a problem with their aorta. Their aorta is weak, and as they age, they are at risk for having an aortic dissection, and that's where the blood goes from inside the lumen of the aorta, dissects into the wall, and just tears the wall apart. And 50% of the people die of the acute event. But if we can figure out who has Marfan syndrome, we can prevent all those premature deaths. And instead of it dying in their mid-40s, they can go on to live a near normal life expectancy. So this really got me interested in the genetic basis of vascular diseases because so many of them are easily treatable if you know somebody's at risk. And so I started working on aortic dissection and the genetic risk. First helped to identify the gene for Marfan syndrome, and then went on to identify the genes for aortic dissection in the general population. And we're up to about 20 genes that my lab and others have identified. And this only explains about 30 or 40% of the disease. And we've gone on, we've stumbled across another gene for early onset coronary artery disease and another gene for early onset strokes in children. And so I've expanded my research now to include different areas of vascular disease. I'm really going after these high impact genes where if you get the mutation, you're virtually guaranteed to have the disease, and these vascular diseases tend to be early onset and a young age of onset. So my lab is sort of divided into multiple different areas. There's a group that does all the clinical characterization and recruitment of patients and their families for a study. There's another group that oversees the sequencing and genotyping of all these families. And then we use a whole number of different techniques to identify novel genes for different vascular diseases. And then once we have the gene, we really want to understand that link between the mutant gene and the end disease that we see in patients. For the students and postdocs entering my lab, the vast majority of them work on this question. This is a question that really gets after the molecular mechanism connecting the gene defect to the actual disease. And right now, we primarily work in mouse models and then get induced pluripotent cells either from the patients or more recently, we've been using CRISPR-Cas editing to introduce missense mutations into those cells and using those two systems to understand how the genes trigger vascular diseases. And the work that we've done is translated back to the patients. So I've got arrows going back to the patients in our study. All the genes that we have identified, and I think we're up to like almost 20 novel genes, are on diagnostic panels worldwide now, and they're used to identify people.
You have 30 seconds, by the way.
Thank you. Before they have to prevent premature vascular diseases. I have my lab, there's currently 4 graduate students, and one of them actually got a very good score in our F30 application recently. And then I have 6 postdoctoral fellows in the lab. So it's a fairly busy and active lab. So I'm happy to answer any questions.